Hi, welcome back. This is part two of Introduction to Genetics. We're continuing on and talking about some of the stuff that you may not have covered in Gen Bio. I want to talk about a little bit about biotechnology, genomics, proteomics, bioinformatics. This stuff may be new. I don't really expect you to have had this except for maybe a gloss over in Bio 1. I want to talk about some of the big things that make biotechnology or sort of the modern understanding of how we can manipulate genetics worth it. One of the biggest advances is the understanding of restriction enzymes. So restriction enzymes are enzymes. They are proteins that make reactions happen either faster or within the lifespan of the organism using less energy. Restriction enzymes are actually used by bacteria to cut viruses apart so that those viruses can't attack the bacteria. Bacteria are constantly under attack by viruses, and this is one of their few defense mechanisms. That virus injects its DNA into the bacteria, and the bacteria releases an enzyme that chops up the viral DNA. It's the chopping up that we love. Because we take these restriction enzymes and we use them in lab procedures to cut DNA that we want to cut at a specific combination of nucleotides. Maybe it's AAA, CCC, AAA. We use a restriction enzyme that grabs onto that AAA, CCC, section and then cuts the DNA there. When we do that, we can either insert a new piece of DNA and allow that organism to now access that new piece of information, or we can cut out something that we don't want. This restriction enzyme technology today we use vectors, or we carry those DNA molecules. We can insert them in a lab situation or in vivo into a living egg and make copies of things. This is how Dolly the sheep was cloned in, I think it was the late 1980s, early 1990s. This is Dolly, and this is Dolly's baby, Bonnie. Dolly was the first mammal that was cloned. That means she's an identical copy of her mom. That's normally not possible in mammals. Normally you need a sperm and an egg. But with Dolly, she has all of her mother's 100%, both sets of her mom's genetics. And she is, behavior-wise, her own individual, but genetic-wise, her genome is exactly the same as her mom. She had Bonnie through more conventional reproductive methods. Bonnie is the result of half Bonnie's dad and half of Dolly's. If you think about, Bonnie is also half of her grandmom because her grandmom and her mom are exactly the same genes. Recombinant DNA technology has allowed us to take our understanding of the shape, the structure, the function of DNA and really manipulate it in a way that is, frankly, amazing. We are going to do this experiment. We're going to take some bacteria. We're going to insert using a series of steps. I'm going to teach you the whole thing as we do it in lab. You're going to do the setup and the experiment and sample it at the end. We're going to take some bacteria. We're going to insert in a glow-in-the-dark gene, and then you're going to let your bacteria grow, and we're going to be able to see who of your bacteria inherited the gene that you spliced in. We're going to genetically modify these bacteria. They're a GMO. They're a genetically modified organism. We're going to create a recombination of genes from a glow-in-the-dark sample and genes from a bacteria that did not glow in the dark, and use a recombinant bit of DNA technology to insert that gene into these bacteria, and you'll be able to see with your own eyes that you actually did that. This is called the P-Glow experiment. You can look at your syllabus. The syllabus, so I know I talked to you the first day. Depending on how those experiments go, we may have more or fewer, but this one we will definitely do. This P-Glow experiment is a lot of fun. You'll be able to really see it. How do we use these in agriculture this week? So it's January, It's January 13th to me. This week there was an article in the paper about gall-safe pigs. So there is a tick, this is a real story, there is a tick in the northern United States that when it bites you, it triggers an immune reaction that causes you to become allergic to meat. This is all meat, so it's not fish usually, but it is you're becoming allergic to an amino acid that is causing an immune reaction. They're just successfully bred pigs that are called gall-safe pigs. They don't trigger the immune reaction in people that have already acquired the immune problem from a tick bite that causes them to be allergic to meat. They're just synthesize their proteins are a little bit differently. These are aqua bounty salmon. They grow twice as fast as regular salmon, producing not as much waste, so they're less environmental damaging, but still producing salmon. BT corn is something we'll talk about in class too. This is a Bacillus thuringiensis. It's a type of corn that has Bacillus thuringiensis inserted in it. Bacillus thuringiensis is a bacterium that when insects eat it, it damages the insect's gut and it kills them. We splice that into a lot of our feed corn so we don't have to spray it with pesticides. It's kind of a pesticide GMO corn. In medicine, we absolutely use gene therapy, inserting genes. We grow things like insulin in labs in bacteria, so we don't have to farm it in an organism like a pig or a human. We can actually grow a lot of insulin in a lab by splicing those genes into a bacteria and then pulling human insulin out of bacteria. We use them for producing things like vaccines, but also for producing hormones and proteins that we use in modern medicine with I guess, from my point of view, a lot less expense and cruelty involved. If you're just farming bacteria, it's a lot less ethically constrained than if I was trying to, make people grow insulin for other people. That would be terrible. How do we apply this? The human chromosome sets are pretty well understood. We understand what genes are on what chromosomes. You have a project that you're going to work on as part of this class where I'm going to ask you to write me a paper or do a presentation, your choice, of a human mutation, so a gene that has mutated. I know that I'm asking you to look at a lot of information. This image here is in your textbook, but I'll also put the entire list of all of these things up on a form on Canvas. If you can't have one that you have your heart set on a genetic disorder, this may be a place to start looking. We know where many genes are that cause genetic disorders, and we know because of the Human Genome Project and the Human Cancer Project, we have a pretty good idea of what it is supposed to code for versus what errors in the code cause the kind of problems that lead to genetic disorders. This understanding has let us treat diseases and anticipate diseases, predict if somebody's going to have them in a way that 100 years ago would have been completely imaginary. So we have a whole bunch of expanding fields here that we're going to talk about, and we're going to do some labs from some of this different stuff. Genomics is the study of structure and function, the evolution of the genes and genomes. This is where we're going to start the class. But we also are going to have a chance to look at some proteomics. So this is proto as in protein. When we look at the expression of proteins in a cell or under a certain set of triggers, do I turn them on, the production of proteins on, or do I turn the proteins off? What do those proteins do? What does turning them on and off at different times in your life cycle or on different cells do? That's proteomics, looking at the expressed proteins. Bioinformatics is, and this may be why you're here, a lot of times there's 2 or 3 students that are really here for the bioinformatics, using technology to now read and understand it. How do I understand the code? How can I process that data? How can I visualize something that is a tiny molecule inside your cell coding for something other tiny molecule? How can I actually visualize that in a way that makes sense to a human brain? That's bioinformatics. We also look at a lot of model organisms. In our class, we're going to start with corn. Corn's a really easy biological way to look at genetics, but what you're probably really interested in is people. But humans are super difficult to experiment on. Why? Why can't I just go out and, I could get you all. I could just do your genetics any way I want. Mendelian genetics, sure. Make you all breed together, make babies. Oh, yeah, see, your red flags are going up really fast, right? Humans are complicated. And I don't mean well, I do, I guess. Relationships are complicated, humans are complicated, we got a lot going on. But a big issue is our genetics are really complicated, and more so than something like a pea plant. There's a huge number of ethical constraints about experimenting on people. We will talk about biological ethics a little bit, but you can't just make people do what you want. Not only should you not want to, probably you're not supposed to do that. And yet, so I need an organism if I want to experiment on genetics, something that doesn't have complicated ethical constraints. Humans are super slow to breed. I mean, you gotta wait till you're an adult and you're like 20 to have babies, and then those babies have to grow up another 20 years. And our lifespan is only, let's say you live 100 years, you can only research a few generations within your own lifespan because our lifespan is the same as humans, because we are humans. That means that I can't do a lot within the lifespan of a single human understanding how heredity works. We live a long time compared to us. We live just as long as our research subjects. That makes it difficult to research them. We really have very few offspring. Most people do not have 10,000 babies, like a fruit fly or a pea plant, which means I can't easily see all the possibilities. I can only see one or two possibilities of combining two people, which isn't great for genetics. So what makes a good organism? Because people are elephants, orcas, they're all terrible research organisms, but they're really interesting. I have to do my research in an organism that is a better model, something that I can reasonably understand in my own lifetime. Mice, fruit flies are sort of famous genetic model organisms. They're not as complicated. Mice are pretty complicated. So are fruit flies, but they're not as complicated as us. There's no ethical constraints or minimal ethical constraints. There's a difference between researching mice and fruit flies. Fruit flies have no restraints on them. Mice, absolutely, you have behavioral and ethical constraints on their research. They both breed really, really fast. If you will have two mice, you can have a lot of mice. If you've ever had mice in your house, you have a lot of mice in a short time. Fruit flies too. Two fruit flies in 8 hours, you got 1 million fruit flies. They don't live very long. They breed fast. They don't live long. I can see the outcome of my experiment many times within my own lifetime. They have a lot of babies, mice, and fruit flies. And I want you to think about some of the other model organisms that we are to talk about and others that you have seen. If they have these requirements, they're probably going to be a good model organism, something that I can understand. And then if I can look at their DNA and watch generations and manipulate generations of them, I can start to find patterns that I might be able to apply to humans or elephants or orcas as well. We're going to look at yeast and bacteria. These are E. coli, that kind of famous bacteria in your gut. I have baby zebrafish here. We don't have them in the lab here, but zebrafish are sort of one of the ones we're going to talk about when we talk about some of the experiments. Super cool. Paper just came out, maybe a year ago. Baby zebrafish not only have no ethical constraints or very few on their research, they also have a lot of behavioral things that are interesting. They actually can count each other very early on. Counting is something that's a big deal. Not all organisms can do it. So you can do a lot of behavioral and genetic experiments on them that then can be scaled up and understood a little bit better about how humans actually work. Yeast, bacteria, nematodes, which are little white worms that live pretty much everywhere. Viruses, zebrafish, all of these are good model organisms because they don't have the problems in research that humans, elephants, and orcas and other long-lived, complicated, slow-to-breed things have. So, much shorter video than the first one. This is new stuff that I think you have not reviewed yet. It's stuff that maybe you don't know from Bio 1. We are also going to use this in the first couple lectures. Come to lab this week if you're watching this. Before you come to lab, if not, I hope you had fun at lab. They will see you on Thursday and Tuesday of every week. In lab, we will start to apply some of the things that we're learning in lecture, and I will expand on it. Our labs starting after week 1 are it says corn lab. We're going to do Mendelian genetics with corn. So we are going to look at monohybrid, dihybrid, trihybrid, and then chi-squared analysis. Those lectures are posted. They are not due until beginning of February for you to finish them and do the homework and everything, but you might want to take a look at them. Again, go over your Bio 1 notes, review mitosis, meiosis, and Mendelian genetics before you start those lectures. You already know some of this. We're just going to expand on it. It's just going to get more interesting and kind of cooler. And again, ask me if you need anything, email me. I'm happy to work with you. I'll see you in lecture. No, I won't. I can't see you here. I'll see you in lab. You'll see me in lecture. And then please ask me if you need any help. All right, I'll see you soon. Bye-bye. So real quick, I was thinking about the ending there and what I ask you to do, and I think I have a little bit more for you. I think my suggestion to you would be to end each video with a little bit of self-reflection. So what are you going to do now? Look at your notes. You just probably took notes, I hope, or at least started to take notes. What words are not familiar to you? What is familiar to you? Is there anything that doesn't make sense? What was the point of all of this? Why did I just do that lecture for you? What information do you think you need to go on? And what do you think if I was going to ask you a quiz question? What am I going to ask you about from that lecture? Now, go to the textbook. I'm going to show you in the next 2 or 3 slides here real fast how to get to the textbook, because sometimes it's weird. Go to the textbook and skim through it. You do not have to read it, but you should look through it. Get yourself a cup of coffee, a cookie, sit down and review through it. See if all those words still make sense. Were they defined in the way you need? There are, at the end of every chapter in the textbook, practice questions. There are regular ones, and then there are extra spicy. See if you can do some of the regular ones. Take a stab at extra spicy and see what you know. I'll go over them with you. Don't worry. We can practice them, and I have, as we go through the course, I have a bunch of different practice with me segments, and we'll do them in lab as well. Do you understand the homework? Click through it. You have multiple attempts at most of the homeworks, multiple attempts at most of the quizzes. I want them to be a learning experience for you, not a punishment thing. That's not the right word, but it's not supposed to be punishing you for not retaining it the first time. I want you to see what you retained, go back and do it again. Look at your notes. Ask me questions, review, and confirm everything that you're doing. When you log in, you might be pink. I don't know if yours looks a little bit different, but you can see on our page, if you go to Access Pearson and you're already accessing the textbook system, if you're not, there's another video for that. You're accessing the textbook system, you click on Access Pearson here and it opens Pearson. So click here, and this is a bit tedious. When you click here, it opens up this view. Go down to Pearson eText. Don't worry about this right now, but go to Pearson eText and click Pearson eText. And it's going to open another window. And then don't worry about this stuff. This is some other person lecturing, and frankly, she's a little bit dry. But it's good stuff. You can watch these videos again with somebody else, but you don't have to. Go here. Go to the e-textbook on the side, and if you click Open e-textbook, it'll remember the last page you were on. Click. And then it opens the textbook itself. The textbook chapters you can navigate by clicking here. If you click here, you can choose Chapter 1, Chapter 2, and the subsets of them. I've numbered my slides so that they match the chapter headings in the textbook. At the bottom here, you can click from page to page. Oh, look, the zebrafish and nematode worms. Oh, these are peas. So very relevant to what we talked about. You click down here. If you keep on clicking, it'll take you to, at the end of chapter questions. There are practice questions for you there. I will also show you in lab if you haven't had a chance to come to lab yet. I think it's worth your time to fiddle around with this. A reminder that a 200 level college course is expecting you to spend 4 hours of your own time studying for every credit of the course. This course is 4 credits. That means you're expected to spend about 16 hours outside of class studying genetics. That's a heckin' lot of time. What I suggest is you do it in little breaks. Watch some video, have a cup of coffee, do something else, come back, fiddle through the chapter. When your brain starts feeling foggy, walk away. Human brains are not meant to learn all the information all at once. Human brains learn by repeating the same experience and developing a framework for what is important. Give yourself little breaks. Spread out your time to study. Do not try to do all this. If you're watching this the night before the first exam, not much is going to go into your head. Hopefully you're watching these on a pretty regular breakdown and spending some time reviewing, and then ask. Let's talk about it in lab. Let's review, so that by the time you get to those exams, it's almost silly, the kind of questions I'm asking you, because you are so familiar with the topic that you can easily answer them, stretch, and go home. And we'll talk about that as well. I will see you in the next video. All right. Bye-bye.